So briefly, we're not going to go too far into the genetics here, but we need to know a few features. So a chromosome. What's a chromosome? Well, in this pictured up top, we see a chromosome. This is referred to as an X chromosome because it has that shape. And inside of that chromosome, we have tightly wound DNA. So a chromosome, we want to say, is simply a unit of DNA. And we inherit a certain amount of chromosomes from mom and the same amount from dad. So we get 23 chromosomes from mom and we get 23 chromosomes from dad, giving us a total set of 46 chromosomes. So answering this question, how many chromosomes do humans have? We want to be able to say 46 chromosomes. Looking at those chromosomes, and this gives us some insight into how change takes place, and it doesn't fully explain, for example, sex from a biological position. But if we look at genetics or chromosomally, and I think at this point of time in science, we can talk about different types of sex. We can talk about chromosomal sex, we can talk about a skeletal sex, and how an individual identifies what a sex they identify with. So just on that chromosomal level, the 23rd chromosome is responsible for that initiating the differences between males and females. It's often referred to as the sex chromosome. So males in contrast to females on that chromosomal level, here we have an X chromosome and a Y chromosome. X because it has that X-type shape, and Y, you'll note that it's notably smaller than the bulking information that the X chromosome has. So it's that XY pattern that's responsible for producing a chromosomal male. And in anthropology, we use a triangle to indicate males. We don't like to use the Mars and the Venus. And in contrast, an individual that's born with a 23rd X from mom and a 23rd X from dad is referred to as a chromosomal female. Like I said, this is pretty simplistic here because in anthropology, when we go a little bit further, we're going to say in some situations, the skeletal sex doesn't align with the chromosomal sex. And whether or not either of that aligns with the way that a person identifies themselves. So that's an entirely different topic that we won't be talking about here. But just so you're aware, this is highly complex, and there's a lot to consider here when we're talking about distinguishing a male from a female. So it's within our chromosomes that we're going to find our genes. And there's a lot of genes inside of a chromosome. If we could stretch out all of the genes that we're going to find in one chromosome, that's going to give us about 8 feet. Now, if we could take all of the genes that are in the human genome, one individual, and we could stitch it all together, it would take us from the Earth to the moon and back about 8,000 times. So there's a lot of information here. Genes represent a portion or a loci of this strand of DNA that's tightly wound up inside of a chromosome. And a gene's responsible for selecting a particular or expressing a particular feature. Let's say stature, hair color, eye color. A gene, then, we want to be able to say it's simply a segment of DNA. That's what gene is, and it's responsible for expressing certain features. And we'll get to how those features are, in fact, expressed. If we look at humans, we see that we do, in fact, have different features. And those features are shaped by genes, but variant forms of a particular gene. So we can talk about something that we refer to as an allele. An allele, so here we've got a gene, and let's use eye color as an example. So we have a gene that's selecting for eye pigment. It's saying that there's going to be pigment in an eye, and that's the feature of this species. So we have an allele for brown. That's what the selection is. As pointed out, we don't see one particular type, we see a number of variant forms. And this is something that Darwin had talked about. We do see that variation, and that's part of this natural selection. So we can go ahead and we can add a couple of other alleles on that particular gene. So we've got one for brown, we've got one for blue, and of course we've got one for green. So we want to be able to say, recall that an allele is a variant form of a gene, and this emerges, let's say, in a particular area. So let's take a strip out of the center of this particular gene, and something different has happened there. There's been a variation in that code. So for example, in the first gene, we've got a collection of A, C, T, and G. And if we just slightly rearrange that in the next allele, we got A, T, C, G, a variant form in that particular gene, and one more time along towards the bottom. And these are just arbitrary here, but A, C, and G and T. So we can see in that selection, it's all selecting for something that's slightly different. And this works for eye color, albeit eye color is fairly complicated. There's a number of genes. But even if we look at skin color, for example, and skin pigment, we're looking at one particular gene, the SLC24A5 gene, that's responsible for selecting dark skin to lighter skin and pigments in between. We like to look at evolution within the concept of genes and alleles. And there's two different types of evolution. There's something that we refer to as microevolution, and this refers to changes that we see in allele frequency over a few generations. So for example, if we see a general shift in the amount of individuals that have, for example, brown eyes, right? Say, argument's sake, let's say 80% of the population has brown eyes, 15% of the population has blue eyes, and 5% of the population has green eyes. If we see a change in this frequency of 80 to 50 to 5, and say in the end we have something like 70 to 20 to 10, we're talking about microevolution. Something's happening to cause this change in the population. Why are we seeing fewer people with brown eyes? Is there an environmental factor that's shaping that, or perhaps even a procreation factor? What's going on? In contrast to microevolution, we have something that's called macroevolution. And this refers to the type of evolution that we're talking about over millions and millions of years. For example, evolving from something that looked very much like a chimpanzee to what we look like today. So we want to call that macroevolution. So with respect to change, Charles Darwin, he had talked about this process where species changed over long and gradual periods. And as pointed out, he couldn't discuss what was taking place from a genetic level, but he did have some idea that new species were in fact being created from this process of change. So today what we refer to it as is a process of speciation. So small changes, they're not going to produce new species. And over time, it'll lead to changes that we see, for example, among the human species. So if we look at the first model here, the anagenic or anagenesis, the anagenic model, we're seeing a unidirectional type of change. So for example, we have ancestral species among humans that might have had slightly different appearances, but they ultimately constitute the same species and the same race. So we can use this currently right now looking at the human species. Traditionally, the groups of people that people refer to as different races, well, in anthropology, we understand that there's only one race, the human race. And within that race of humans, we do have individuals with different ancestries, different areas of the world that they've come from. And there are certainly biological, genetic, and physical differences that exist between us. But we all belong to that same species, and we're changing over time. And mainly this is because we're engaging in something that we call gene flow. We'll talk a little bit about that later on. In contrast to the anagenic model, we have another model that we call cladogenesis, or the cladogenic model. And this is where we're seeing that process of speciation. So usually there's going to be some sort of event, some geographic boundary, something happens that separates two species and puts them on different evolutionary courses and leads to that process of speciation. So here in the second model, B, we see two different species emerging from this ancestral species, B, in the cladogenic model. And they've broken off at that green dot that we see, and they've gone on their own separate evolutionary path. Now, over time, as we see changes in allele frequency, that microevolution, that's going to start to add up over time. For example, in the cladogenic model where we see species B, we're seeing a lot of change taking place, and they're not interbreeding with species C. Now, species C, they're undergoing their own changes, so these species begin to drift apart and lead to two different species eventually, but with a common ancestor. As pointed out, Charles Darwin could have benefited from reading a little bit of the work that Mendel was engaged in here. Our textbook covers it, but we'll go over it briefly. So Mendel argued that heredity is determined by small particles located on chromosomes, and it's those chromosomes that transmit genes across generations. So Mendel offered this law of segregation, and what the law of segregation states is two alleles are inherited for any given gene, one from each parent. So, for example, if we use eye color, and I'm going to eye color because it's something we can relate to, but in fact, eye color, it is quite complex. You might have blue eyes because you've got broken brown eye genes. There's a number of explanations here, but we'll just take a look at this to get a basic understanding. So Mendel said that these alleles, they can be homozygous or they could be heterozygous. Now what he's getting at is, for example, if we look at eye color, say you've got a brown allele from your father and a brown allele from your mother, you are homozygous. You have the same alleles, and you're going to have brown eyes unless one of those brown eye genes or both of them are broken. In contrast, if you've inherited a brown allele from your father and you've inherited a blue allele from your mother, then you are heterozygous. The other component is the alleles can be dominant or recessive. So dominant alleles, they cancel out recessive alleles. And the only way that recessive alleles are expressed is if you are homozygous for those recessive traits. I'll come back to this in a minute to help you understand it. So a couple terms. We want to say homozygous, chromosome with identical alleles. And in contrast, if something's heterozygous, the chromosome has different alleles. Now, the thing to remember about dominant and recessive is just because you say you have a recessive gene, it doesn't go away. It's still there. So what we can say is that the dominant alleles, they can mask recessive alleles, and they can come up at a later point of time. You can pass them on because they're still there. So let's take a look at this in another way. We could pose the question, just because things are masked or hidden, do they go away? And I'm going to answer, well, just because things are masked or hidden, it doesn't mean that they go away. They're still there. So, for example, just because this dog is hidden under the blanket, Does it go away? No, of course not. It's still there. And then, of course, the cat under the blanket, it's hidden. It will say it's recessive. It can't be seen. But does it go away? No, it doesn't go away. And then, of course, we have the pigs in a blanket, and well, they go pretty quickly. The point here is just because something is hidden, it doesn't mean that it can't emerge at a later point of time. So Mendelian genetics then is looking at inheritance patterns. It's interested in whether or not an individual is homozygous or heterozygous for a particular type of allele, and how those alleles either cancel out each other and what they produce in the end. So allele dominance then, it creates a distinction between our genetic makeup and our physical characteristics. And they can in fact be different. For example, somebody can carry a blue allele for eye color and not have blue eyes. So you can take two individuals with brown eyes and they can logically have a child with blue eyes because they both passed on recessive genes. So I'll show you how this works. So let's say we have a genotype, three different types of forms here. So the genotype or the genes that an individual has, you can be homozygous for tallness, we'll say, capital T. Or you could be heterozygous, where you have a gene for tallness, capital T, and you have a gene for smallness, a small t. Or you could be homozygous for small, represented by two small t's here in our PowerPoint. The phenotype in each one of these situations, it's going to be expressed as such, unless there's a broken gene. So in a situation where an individual is born with homozygous for tallness, so two capital T's, that individual is going to be tall. And this is what Mendel was doing with plants. So he found tall plant plus tall plant. With those two genes, they're going to produce tall plants. When you crossbreed, for example, a tall plant with a small plant, he found, well, tallness is expressed. So in this situation, the capital T, it's dominant, and it's canceling out that recessive allele for smallness. When we cross or we breed, for example, two small plants, TT represented two small t's here, the shortness is going to be expressed. Moving away from Mendelian genetics, which deals with how genes are inherited or transmitted across generations, anthropology and evolutionary biology is also interested in population genetics, or the mechanisms of evolution. How does all this stuff take place? So here I want to talk about four different types of mechanisms. I want to talk about natural selection, mutation, random genetic drift, and gene flow. We're familiar with natural selection. We said that this is a process whereby nature selects for the fittest species, and Darwin and Wallace offered this. The image here that we're looking at, we see a bird of prey with three different types of moths in each square. The first one, we see two light-colored moths on a similar surface, tree trunk. They're camouflaged. Below them, we have a black moth, and on that lichen covered bark, it's not at all camouflaged, and it's going to be less successful in that environment. So here, nature selecting for the camouflaged species. They're not getting eaten by the bird of prey, and their reproductive success increases. And that's kind of the other key point. We need to think about reproductive success. That's the way that evolution works, through reproductive success. In contrast, Following the Industrial Revolution, at least the argument goes, the soot produced from the factories, it started to cover those lichen-colored trees. So we see a different type of relationship and a different type of selection. So now the light-colored moths, they're very visible on a soot-colored tree, and the bird of prey is going to eat them, whereas the reproductive success of the dark-colored moths, it increases in this particular environment, and they're fitter. Now, everything works through natural selection. That's the main overarching principle of evolution. But there's a couple of other mechanisms through which change takes place. And perhaps the most important, or the only way that we get new genetic material into a gene pool, is through mutation. So first, let's talk a little bit about gene pool. I want you to think of a gene pool as all of the genetics that are available to a species, for example, the human species. So in anthropology, we do not like the term race. There's one race, the human race. There isn't, we don't like to talk about African races or European races because we're all so close genetically. There's not enough of a difference between us to justify using that type of language. Like I said, I'm going to say this a few times in our lectures, we are 99.7% genetically identical. Doesn't make a difference where we come from. So if we want to compare that idea that a gene pool is all of that information that's available to us genetically, let's think of Walmart. That's kind of like a gene pool. Everything, last time I remember, I haven't been into a Walmart in a very long time now under the physical distancing. But either way, when I did go into a Walmart, I could buy tires for my car there. I could buy a computer. I could grab some apples, some eggs. I could buy some clothing. I could buy a magazine. I could get something from McDonald's. I don't eat McDonald's, but I could get something from McDonald's. I could buy a magazine and on furniture. Everything is available. So that's kind of like a gene pool. All the genes that are available to the human species. Now, change or new genetic material, the only way that it can be introduced into a population is through mutation. Mutation is defined as a random change in genetic material. It's a copying error. And you know, we do have these mechanisms in DNA. It does have a mechanism where it tries to correct for some of these changes, but it's not perfect. We don't have a perfect correcting mechanism all the time. So sometimes slight changes emerge. That's what Darwin was talking about. He couldn't use that term mutation because it just didn't exist at that time. But either way, through mutation, we get potentially new genetic material. Now, we tend to think of mutation as something that's bad. We think of a mutant. Mutation, it doesn't necessarily need to be bad. When we look at, for example, like I said, light-colored skin, that's a mutation. We look at blue eyes, that's a mutation. We look at green eyes, that's a mutation. So the ability for humans to walk upright, all of that stuff fits into that category of a mutation. In some situations, and here we're looking at the SARS-CoV-2 virus, mutation can be quite worrisome. So that new genetic material, for example, if we're talking about viruses or bacteria, it can become potentially harmful. If it allows it to, say, increase its infectivity, it's going to get into us. That's increasing its success. So depending upon the species we're looking at, depending upon how that change is impacting the species, it could lead to the eradication, for example, of the virus. Or it could lead to its increased success in the fact that it may never go away. What we could expect to see, I would imagine, my experience with looking at studying disease, is the virus will become weaker over time, kind of like the cold virus. Believe it or not, in the late 1800s, there was an epidemic that killed millions of people. And what we know about it now, the virus that was responsible for that pandemic that killed millions of people, is one of the cold viruses today. It became weaker. We catch it, we get grumpy, we get a stuffy nose, we sneeze, and over time it goes away. So that's part of that process of natural selection as well. But mutation, random change, genetic material. And as pointed out, this is the only way that new variation, genetic variation, can be introduced to a species. So the third mechanism is random genetic drift, and this is best understood as a sampling error. And while you think that if I flip a coin, and I have my lucky coin here, if I was in class, I would have it today. It's an English pound with a Neanderthal on one side from Gibraltar, and on the other side, Queen Elizabeth II. And we can hear it there. Okay, so, if I flip this, say 10 times, what's going to happen? We're going to try it right now to prove something. So I'm flipping it, and I've got a Neanderthal head. I'm flipping it, and I've got a Neanderthal head. 3 times. Okay, queen, so 2 to 1. Neanderthal head, that's 3. Flip again, Neanderthal head, believe it or not. And Neanderthal head, and of course, a Neanderthal head. And we can see, and I'm not going to continue on with that, but we can see what's happening here is it's not balancing out the way that we'd expect. I'd expect to get 50% Neanderthal heads and 50% Queen Elizabeth II heads, but that's not the case. That also happens with genes as well. So random genetic drift, it refers to a change in allele frequency as a result of chance. The thing to remember, now flipping this coin, when I do it 10 times, I have a greater chance of getting Neanderthal heads or Queen Elizabeth heads at a disproportionate ratio. More Neanderthal heads to Queen Elizabeth heads. As I stretch that out and I say I flipped it 100 times or 1,000 times, I'm going to have a greater chance of getting that 50-50 balance between Neanderthal and Queen Elizabeth. So what we want to say is we got a greater chance of one allele becoming fixed in a small population rather than a large population. So here's a little experiment to try to illustrate this idea. So what we're seeing here is a number of generations. You can do this at home with Skittles if you don't eat them all, but you want to start out selecting your Skittles, red to blue, say 50-50, put 50 red in, 50 blue in, and begin to take those out and say the first time I have 100 Skittles here, I'm going to take 50 Skittles out and put them in the next jar and see what the ratio of red to blue is. And what we're seeing is a change. So we started out 50-50 in the original population. Second population, we're seeing fewer reds. Third generation, we're seeing even fewer reds. The fourth generation, what's going to happen here? We're almost there. Doesn't want to select. Oh, 4. And the last generation, we've gone to an equal amount. So we could do this a number of times. And what we're going to see is we're not always going to get 50-50. Now, if each one of these marbles or Skittles or Smarties represented a feature, as we're starting to move through time, it's quite possible that we might see only one particular type of feature represented, or at least represented dominantly within a population. The interesting thing about random genetic drift is it also involves something that we call the founder effect. Okay, so here we've already got this. We've got refers to a change in allele frequency as a result of chance, greater chance of one allele becoming fixed in a small population rather than a large population, and we've got something called the founder effect. So with the founder effect, what you need to imagine is a few people leave that population, where perhaps we saw an even distribution or whatever the normal distribution of brown to blue to green eyes would be. A few people leave, and they leave, say, without brown eyes. There's nobody in those founders that have brown eyes. And they move to an island or a new area. We're going to see a dominance of perhaps green and blue eyes, or maybe just blue eyes, whatever emerges. So with the founder effect, what we want to say is the isolation from the parent or other populations, parent population or other populations, cause the frequency of the founder's genes to be commonly expressed within the population. So looking at populations where we see a dominance of red hair, that could be a result of this founder effect. Our last mechanism of genetic change is something that we refer to as gene flow. Now, gene flow prevents speciation. And gene flow, in the end, as the kids are saying now, Netflix and chill. That's what it is. It involves intercourse, and so long as populations are having intercourse, interbreeding, we're not going to see speciation take place. We're going to share genes. We're going to share those new mutations that emerge, and we're going to continue to be the same species. And this is why we can say the human species is just that. We are one species. So gene flow, we can define it as an exchange of genetic material between populations of the same species. And as I've pointed out, it prevents speciation. So here, looking at the image, you know, if this mountain range wasn't here, it would be a geographic barrier, right? If we had a barrier right here, and it wouldn't allow these the western deer population to breed with the eastern deer population. So as new mutations emerge, for example, among some of these species over here, those mutations are going to stay within this population, and over time they're going to go through that model that we've talked about, that cladogenic model, I'll draw it up here, where they shared ancestry at one time and they break off, and they eventually can become subspecies and then eventually different species. But because we have a pass between this mountain range, it allows the brown eastern deer population, lighter colored, here beigeish, to interact with the goldish western deer population. So any mutations that take place within this population are going to be shared with this population through that process of gene flow, and vice versa, and they will continue to share that gene pool.